So before we move on to the other mechanisms of making a fetus and an embryo from a single zygote, I just want to come back a little bit to one aspect of the cell cycle, and that's this DNA synthesis phase. Like I said before, this is where the DNA is getting copied. If you look at it, what's happening is this. In G1, the DNA is this long single strand of material that we've seen before, and this is what it looks like in an electron microscope. Before we go to mitosis and meiosis, the chromosome is going to condense, and you notice when it condenses, you end up with these sister chromatids, which we talked about before, connected at the centromere. There are no sister chromatids, however, in G1. In G1, there's only one long strand, essentially one chromatid. It's in S, it's in this phase here, that we get the sister chromatid. This entire chromosome copies. And when it copies itself, now you have these two identical copies, which you can see here, and they are connected right in the middle in this centromere region. So just before M, just before we go to the M phase, or actually at the beginning of the M phase, in a part that's called prophase, the chromosome goes from this open sort of configuration to a condensed. It curls up and coils on itself to make this condensed chromosome with the sister chromatids. So in G1, this is a cartoon of what's happening. You notice all the chromosomes are just single strand, one strand of DNA. Then in S phase, they get copied. And so here in G2, you're going to have now the chromosomes that you're used to seeing in the traditional textbooks. They look like this. Then during mitosis, what happens is that these cells go through prophase, prometaphase, metaphase, anaphase, telophase, and give rise to then two new cells. And when they do that, these chromosomes get ripped in half, and one chromatid goes to one cell, and the other chromatid goes to the other cell. I'm not going to spend a lot of time talking about that in lecture because that's going to be something that we're going to deal with in very great detail in the laboratory, and there's no point in doing it twice. But realize, I do want you to connect that laboratory portion of the mitosis-meiosis laboratory with this section right here, exactly what is happening during mitosis to divide that nucleus. Okay, so like I said, you start out with a single cell type, a zygote, and yet you end up with how many cell types at the end? You go through this body and you make this fetus, for example. How many cell types are there when you get to this phase right here, the fetal phase? Turns out in a human being there's about 200, approximately 200 different distinct cell types, and they all came from this one zygote. So how did that happen? It isn't just a matter of making a bunch of new cells. Well, that's another process, and that process is called differentiation. The word differentiation means changing form and function, in particular of a cell, as it develops. So for example, let's take a look at what are called stem cells. Now stem cells are something that you have almost certainly heard about, and if your understanding of stem cells comes from the journalistic media, your understanding is undoubtedly incorrect. Stem cells are cells that are capable of becoming other types of cells, but they also have other properties. Specifically, they have this property that's called asymmetric division, which we'll talk about later in the course. But in essence, for right now, a stem cell is a cell that can give rise to a number of different types of cells in the body. Sometimes they can give rise to all the different cells in the body, in which case we call them totipotent. And cells, for example, in the morula are all totipotent. If you were to take this cell out of the morula and grow it as if it were its own zygote, it would grow perfectly fine into any of these structures and make a complete new fetus. In fact, that's very much how identical twins are born. The morula falls apart, and each one grows into a unique individual. However, by the time you get to the embryo, the cells are committed to becoming something specific, in which case then they can't become all the different cell types, they only can become some. Here's an example of one of those types of cells. It's called a mesenchymal stem cell. And this mesenchymal stem cell is actually also in your body. These mesenchymal stem cells can give rise to a variety of different types of cells. For example, when it divides, one of its daughters, one of the other cells that it makes, can become what we call a transient proliferator. That's a term you'll learn later on. But those transient proliferators will start to change into one of these different main types. So for example, this proliferator could become an osteogenic cell. In other words, it goes through this differentiation pathway and becomes what's called an osteocyte, which is a cell that you find in bone. But the exact same cell could also change into a different type of cell using a different DNA program. It could go through chondrogenesis. Chondro refers to cartilage. And so it could become one of these types of cells, a chondrocyte. It could also go through this process called myogenesis, in which case then it becomes a myotubule or a muscle cell. It could go through the marrow forming process. It could go through the fibroblast forming process. It could go through the adipogenesis process, become a fat cell in the adipocyte. It could go through a series of others. But, for example, this transient proliferating cell could not become a neuron. It could not become a hepatocyte. So there's a number of cells that it can't become and a number of cells it can. But this process of differentiation is how we get the different kinds. Now, the thing you got to remember is all of these different cell types came from this one mesenchymal stem cell. And the process of mitosis, the process by which cells divide, go through this. It causes this to happen. The DNA gets completely copied, and one copy goes to one daughter, and the other copy goes to another daughter. So that means this. These cells are clones. They're absolute clones of each other. The definition of a clone is not just identical, but genetically identical. They have exactly the same genes. So that means this. All of these different cell types down here, all these different cell types are genetically identical. They're clones, even though they look very, very different and perform very, very different functions. Every single cell in your body, except for a very few, the sperm and the egg cells in your body, they are all exactly the same genetically, therefore they're clones. Okay, so that gives us the 200 different cell types that we find in mammalian bodies. So now we have the trillions of cells through proliferation. We have the 200 cell types. And the question then becomes, what about this process? How do you form, for example, a hand, or in this case, the foot of a bird? How does that happen? How do you form a heart, a structure like the heart? How do you form a structure like the brain? That process is another process by which organisms develop, or that they use when they develop, called morphogenesis. The word morph does not, oddly enough, mean change. That occurred in the 1980s. It took on that terminology in the 1980s because it became a movie's special effect. But morph actually means form. Genesis means to produce, so morphogenesis means producing some sort of form. In this case, we're producing fingers that are going to be, for example, in quails or other types of organisms, chickens, mice, and things like this. Your hands develop in exactly the same way. Now, let's go back to the fetuses. If we look here, you see how you got these little tiny blobs here, these things that stick out like this. And you see they're just nubs. Then eventually you end up with fingers. How is it that we end up with these fingers? How does that occur? It isn't that the fingers grow out of the ends of these nubs. That's not how it works. What happens is the nubs end up with these lines, and you can see here a line here and a line here of dark cells, and dark cells here and dark cells here. In this particular study, what the researchers did is they gave the cells a particular chemical that the cell would take up if it was going through a particular genetic program and not if it wasn't going through that genetic program. The genetic program that they're highlighting here, for example, this dark streak here, a bunch of cells are turned on a program that's called apoptosis, or just apoptosis. Now, the word is apo and then ptosis. It is correctly pronounced apoptosis. The word ptosis, the P is silent. Now, you'll hear a lot of people pronounce it apoptosis. That's fine. There's nothing really wrong with that. But the correct pronunciation really is apoptosis. Apoptosis is that program I was telling you about earlier where the cell commits suicide. It kills itself. That's what's happening. These cells here, all these dark stained cells, you see the lines here and here, they're all killing themselves. They're all dying, and they continue to die, and they continue to die, and they continue to die until all of this tissue between the fingers, there's a finger, there's a finger, there's a finger, or toes, all of that tissue is dead, and it separates then into individual toes or fingers. So this then shows exactly one of the main mechanisms, anyway, by which morphogenesis occurs. You have a blob of cells, they start to differentiate, But then this apoptosis process kills away and sculpts the structures that make up the main forms of the body.